Good afternoon. Today I will explain about cohesive force and adhesive force in the xylem using the model that I built using recycled items. The materials needed are scissors, colored plastic bags, recycled papers or newspapers, and tapes. This is how you build the model. First, crumple a piece of paper into a small ball. Then you wrap it with the colored plastic bag. And tap it using and then you use a double-sided tap to tap it on either a manila card or a paper bag. This is the model that shows cohesive force and adhesive force of water in the xylem. Water molecules are attached to each other through a cohesive force. At the same time, water molecules are also attached to other surfaces through adhesive force. Both forces produce capillary action, which allows water to enter and move along narrow spaces, such as in the xylem tube. That's all for my presentation on cohesive force and adhesive force inside the xylem. Thank you.
Hi guys, today I'm going to talk about lock and key hypothesis. So first, what is enzyme? Enzymes are molecules and they are made up of amino acids. So this is enzyme, and if you have a closer look at it, you'll find that enzymes are actually made up of teams of amino acids that are folded up to form a specific 3D structures. Our cell is actually a very busy place, and most of what it does is to move molecules around or change molecules into other kinds of molecule by either making smaller molecules bigger or bigger molecules smaller. Let's see, enzyme react as the lock because each known as enzyme molecule has a region with various shaped active sites. And this is the substrate. The substrate must have a complementary to the active site of enzyme so that they will fit into the active site. Again, enzyme react as a lock, substrate as a key. Only the correctly sized key into the keyhole of the lock. Mean that the enzyme specifically reacts with only one or very few similar compounds. Once the substrate is attached to the enzyme, the whole thing just becomes the enzyme-substrate complex. Once the bonds are broken, the product will be formed. The products no longer fit into the active site and they will escape into the surrounding medium, leaving the active site free to receive other substrate molecules again. So, as you can see here, the structure of enzyme molecules still remain unchanged and you still bind with other substrate molecule for other reaction. Let's conclude the topic. Thank you, have a nice day and stay safe.
Assalamualaikum and good evening. I bid to Madam Shen. Today I would like to represent the subtopic 4.1, which is cohesive force and adhesive force of water. The material that I will be used today is ruler to represent the xylem tube and paper that has been cutted to represent water molecule. First of all, when the water molecule are attached to each other through cohesive force. Cohesive force is property of light molecules of the same substance. That is why when the water molecule attached to other water molecule, it is called cohesive force. Next, when the water molecule attached to other surface, it through adhesive force. Adhesive force is actually property of different molecules. That is why when the water molecule attached to the surface of xylem, it is called adhesive force. Next, the combination of the cohesive force and adhesive force will produce capillary action. Capillary action do not require the force of gravity because it reacts opposite the gravity. The water will move upwards instead of downwards. This capillary action will allow the water to enter and move along the narrow space, such as in the xylem tube. Xylem tube can be found in the plants. That's why the water will move upwards instead of downwards. I think that's all from me. Thank you.
Good morning, Abid to Madam Shem. Today, I would like to present about a topic 4.1, which is cohesive force and adhesive force of water. Materials that I will use for today's activity is a ruler and plasticine. This ruler will present the xylem tube and this plasticine will present the water molecule. From this structure, you can see these water molecules are attached to each other through cohesive force. At the same time, the water molecules also attach to other surface through adhesive force. Both forces are produce capillary action, which allows water to enter and move along narrow spaces, such as in the xylem tube. And I think that's all from me. Thank you. Have a nice day and stay safe.
A few moments later.
Hi madam. So today the topic that I choose is development of human fetus. Fertilization can occur when the one out of millions of sperm succeed in penetrating the secondary oocyte in the fallopian tube. This is followed by changes to the secondary oocyte's membrane that prevents penetration from other sperm. Then, fertilization occurs when the sperm nucleus fuses with the ovum nucleus to form a diploid zygote. While traveling down the fallopian tube, the zygote undergoes multiple division to mitosis. The first cell division produces 2-cell embryo. Then it divides to 4 cells. At cell, wing cells division will finally produce a morula. This is how cell divisions will finally produce a morula. The morula then transforms into a blastocyst. The blastocyst will then implant in the endometrium. This process is called implantation. The thickened blastocyst wall will develop into embryo. The placenta also produces the human chorionic gonadotropin HCG hormone during pregnancy. The level of this hormone increases at the early stages of pregnancy and will double every 2 to 3 days for the first 4 weeks of pregnancy. The main function of HCG is to make sure the corpus luteum continues to secrete estrogen and progesterone in the early stages of pregnancy. This hormone can be detected in the urine of pregnant mothers. So I think that's all for me. Thank you for watching.